Yesterday, guys, we started with chapter 8, section 1. The title of this lesson was Identifying DNA as a Genetic Material. We learned that Mendel found out about traits that inherited. إنه في عندي dominant وعندي recessive, but he didn't find out or study about the chromosomes. Okay? بعدين after him إجا مورغان ومورغان he discovered إنه في عندي linked genes يعني some genes are inherited together because they are very close. تمام؟ But who طيب okay Iyad, I got your message dear. Okay, who identified this material اللي هي هدول الكروموسومات اللي هم عبارة عن DNA. Who identified them? Who knows or who discovered إنه هاي اللي هي DNA هي the material that is responsible for your characteristics, for your traits. هي genetic material. يعني احنا بنعرف هلا إنه DNA هي genetic material. هي genetic material that give your body the instructions that control the making of many molecules like proteins in your body. هي تتحكم في كل شيء in your body. Okay? طيب how they discovered إنه هاي DNA genetic material. ليش ما طلعتش مثلا genetic material protein؟ مش احنا عندنا 5 carbon based molecules، عندنا الكربوهيدرات وعندنا الليبيدز وعندنا الامينو اسيد البروتين وعندنا النيوكليك اسيد اللي هم الدي ان اي، صح؟ مش احنا عندنا in our body we are made of 5 carbon based molecules، صح ولا لا؟
Yes.
احنا we are made of 5 types of carbon based molecules اللي هم كربوهيدرات وبروتين وليبيدز ونيوكليك اسيد. اوكي؟ هلا النيوكليك اسيد هي دي ان اي. طيب how they identified انه هاي الدي ان اي هي جينيتيك ماتيريال، مش كربوهيدرات هي جينيتيك ماتيريال ولا بروتين هي جينيتيك ماتيريال؟ كيف عرفوا؟ هلا طبعا this happened by experiments of some scientists. Let's see how they discovered this genetic material. Okay, and start with this video. The first one كان اسمه Griffith. Okay, let's see what did he found out.
This is our timeline of the history of genetics. So far, we've covered the important milestones up to this point. Yes, we've taken a look at the work of Gregor Mendel, followed by the work of Sutton and Boveri. This was the chromosomal theory of inheritance. Later, it was Thomas Hunt Morgan who put forth his work on the common fruit fly and achieved another milestone. Now comes the next one. After Morgan gave an experimental evidence of the fact that genes for the traits are present on the chromosomes, the scientific world then got together to find out what exactly makes up these genes and chromosomes.
يعني Morgan he found out that the chromosomes they have genes, okay? But what they are composed of, what are these genes? Who found that? اللي هو the first one who
chromosomes. In other words, the chemical nature of this genetic material is what everyone was curious to know about. In the same era,
I wanted to know what is this genetic material? ايش الchemical that make that chemical component that make this genetic material? هل هو carbohydrate؟ هل هو nucleic acid؟ ايش هو هاي الماتيريال؟ They were curious to know.
British bacteriologist was busy handling his pneumococcal strains. They are nothing but the bacteria named Streptococcus pneumoniae, which are the causative agents of pneumococcal infections.
هاي نوع من البكتيريا اسمها pneumococcal strain، في منها أكثر من صنف. نيومونيا هي بتعمل انفكشن عادة بالريسبيراتوري سيستم أو باللنج اسمه نيومونيا. هاي النيومونيا في كان عندها سترينز أكثر من فصيلة. في عندي منها بكتيريا harmful وفي منها harmless، والharmful بتعمل انفكشن. هذا الساينتست كان اسمه جريفيث. He was studying على هذا type of bacteria.
Frederick Griffith found out that there is some factor that helps transform one bacterial type into the other. Let's have a look at this remarkable experiment. To begin with, we need to get introduced to the two types of pneumococcal strains. This one is a smooth strain denoted as the S strain, and this one is a rough strain denoted as the R strain.
اوكي he found out there are two strains. كان عم بيعمل دراسه على two strains من هاي البكتيريا. في بكتيريا كانت عندها الكفر تبع البكتيريا سموث، في كبسول يعني it's covering this bacteria، وعشان هيك عندها الكفرنج تبعها اللي هو سموث، فسماها smooth strain and call it by S. وفي الأذر سترين كان الكفر تبعه رف، شايفين؟ هلا وسماه الار سترين، تمام؟ he found out انه هذا السموث سترين هو اللي بيعمل هو اللي pathogenic. شو يعني pathogenic؟ هو the type that cause infection، while the rough strain is harmless, doesn't cause infection. إذا الاس هو اللي بيعمل infection.
Why are they named so? This smooth strain, as we can see, has a smooth outer surface. It's the capsule around the cell wall which protects it from various threats. The presence of this capsule makes the strain virulent or strongly infectious, to be precise. The absence of such a smooth coat makes the strain avirulent or non-infectious, to be precise.
Virulent, avirulent نفسها infectious not infectious. Okay, هذا cause infection، هذا doesn't cause infection، هذا harmful، هذا harmless. Okay?
You may ask, why is the R strain avirulent? It's the absence of the capsule which makes it susceptible to the immune cells of the host's body.
هلا هي مش مهم تعرفوها، بس for your information ليش ال R strain is avirulent is harmless؟ لأنه ما في كبسول. هذه ال S strain في هون كبسول that covers this rough surface. فالimmune system ما راح يتعرف عليها، it can attack our body. أما الavirulent بيكون على السيرفيس ما في كبسول، فالimmune system when they find this body بيحسوا إنه هذا strange body invader، فبيعمل له fighting على طول. Okay?
Now getting back to Griffith's work.
يس يا عبد العزيز.
انه اذا الاميون سيستم ما تعرف عليها برضه نفس الشيء الرف سترين وفاتت مثلا، فهاي كمان ممكن انه
في مشكله بالاميون سيستم. ممكن بالاميون سيستم بيكون في مشكله، بس عاده الرف سترين الانسان الطبيعي خلص بيهاجمها، عرفت؟ بس هاي لانه عليها كابسول هاي الكبسوله مغطيه، يعني ما بيخلي الاميون سيستم يتعرف عليها، فممكن تسبب المرض الجسم بدون ما يدافع عن حاله. اوكي؟ do you understand؟ alright. تمام. إذا هلا اتفقنا الاس سترين هي الانفكشن أو الفيرولنت، والرف سترين انفيرولنت أو not infectious. تمام؟ شوفوا إيش عمل.
Let's see what experiment he exactly performed. He first injected a mouse with the S strain, which is the smooth strain of the pneumococci.
شو رأيكم؟ أول شيء عمله he injected الماوس with S strain. What do you think will happen to the mouse? What do you think will happen to the mouse, Abdullah?
Teacher, yes.
Yalla.
It will be smooth.
First of all, he injected the mouse with S strain bacteria. What do you think will happen to the mouse?
Will it be smooth?
لما يحط في الماوس الاس strain he will be smooth. احنا قلنا الاس strain is infectious والR strain not infectious. What will happen to the mouse when he inject the mouse with the S strain? He will become infected. Bravo. He will become infected. Thank you.
Yes, I thought, teacher, you were asking what did he get infected by?
What will
smooth
happen?
Strain. Oh, okay.
The mouse died.
I understood the question wrong. Sorry, teacher.
Now, after he injected the mouse with the S strain, the mouse died. Okay? Let's see what happened next.
Similarly, on injecting another mouse with a rough strain, the mouse survived. This was...
So, when he injected the mouse with the S strain, the mouse died. when he injected the mouse with the R strain, which is avirulent or not infectious, the mouse stay alive.
Was pretty much anticipated by him because the rough strain is usually the avirulent one. What was done next? Now the third.
She said a rough strain usually avirulent, usually, but يعني إذا كان في مشكلة بالimmune system لا it will be infectious طبعا. Okay?
Yes.
تمام. Alright, let's see what happened.
Third mouse was injected with the S strain, but after the bacteria were heat killed.
شوفوا شو عمل. هلا he injected the mouse with the S strain after heating the bacteria, يعني after killing the bacteria. He take the S strain, he killed this bacteria by heating. انت عادة لما تحرق البكتيريا خلص you kill the bacteria. تمام؟ ف he injected the mouse with heat killed S strain. What do you think will happen to the mouse? He will stay alive ولا yes يا زلمة؟
لحظة مس كان ask a question.
محمد يس.
مس الهيت كيلد سترين ايش حكينا؟
مش سامع محمد وات؟
مس الهيت سموث سترين حكينا ايش بتعمل؟
انت ليش دخلت متأخر اولا؟ ليش دخلت متأخر؟
مس والله ما بعرف ما وصلني شيء هسه قبل شوي وصل اللينك.
ما هو انت مش حاضر الفيديو من اوله صح؟
لا حاضر من الحصة الماضية.
لا هلا دخلت انت؟
هلا.
طيب اعيد انا عشان تفهموا، اوكي؟ تمام. هلا كمان مرة ثانية بليز ما حدا يتأخر بليز. The first أول شيء he made a study about two strains of pneumococcal bacteria. اوكي نوع من انواع البكتيريا اللي اسمها pneumococcal. هاي البكتيريا في منها أكثر من strain، اوكي؟ هلا في عندي strains بتكون pathogenic وفي strains non pathogenic. استعمل التو سترينز هو. السموث سترين كان بيكون حواليها كابسول، هاي بتكون باثوجينيك. والرف سترين ما في حواليها كابسول، هذه اللي هي نون باثوجينيك او هارملess. تمام؟ First of all حكينا he injected the mouse with the S strain. What happened to the mouse? Died, okay? And the other, then he injected another mouse with the R strain. Which is not infectious. صار ايش صار فيه؟ he stay alive. لهون تمام محمد؟
اوكي.
يس مراد.
أمس عدي أجاوب بالسؤال سألتيه عن what will happen to the mouse when he's infected with heat killed.
اوكي just بعدين شو عمل؟ then he took another mouse. he injected this mouse with a heat killed S strain. أخذ S strain bacteria, but before he injected, he heated the bacteria to kill it. Okay? فهمت يا محمد؟
يعني هاي بكتيريا ميتة؟
ميتة بالضبط. أخذ البكتيريا وهيتد. مراد, what do you think will happen if we inject this mouse with this heat-killed bacteria?
Yes, I think that the body will get rid of it and the mouse will live.
alive ولا it will die?
He will stay alive.
برافو. هذا اللي صار فعلا. When he killed the bacteria and inject the mouse with the bacteria, الماوس stay alive. خلص ماتت البكتيريا. اوكي؟
ماتت. زي كأنه حقنه بمياه.
بالضبط. بالضبط. إذا هنا he stay alive. يعني it doesn't kill the mouse because he killed the bacteria. Yes يا مراد.
Yes, Miss ما عندي سؤال. نسيت.
طيب lower your hands if you don't have any question محمد. Okay. Now, what will happen? What did he do next?
That means.
Yes, Muhammad.
No question.
لا, lower your hand. Only raise your hand when you need to.
يعني عادي أحكي وأنا مش رافع إيدي.
يعني okay بتصير. When I tell you stop, you have to stop. Okay? Now, but while we are running on the video, don't disturb. Okay? When I stop it, we can talk. طيب هلا let's see what he did next.
He heated the S strain to extreme temperatures, which killed the virulent strain and made it ineffective.
شايفين؟ when he heated the S strain كانت تحولت من virulent ل avirulent، من infectious ل non infectious، من harmful ل harmless. Yes عبد العزيز.
بس if we burn the S strain هيك حيكون السيرفيس rough surface.
بالضبط. راح الكبسول
حتى هيك كمان الاميون سيستم.
هم بيقولوا لك هون انه when you heat it خلص it become ineffective زي الرف.
yes.
Yes, any question? Yes, Muhammad.
Miss, does that mean if you have a bacterial infection in your hand, if you heated your hand, the bacteria will die? Like, نحكي في عربي كو أو كواء.
حسب وين الانفكشن، إذا سطحي إذا داخلي، بس طبعا الكوي مش محبذ أبدا يعني. Okay? Let's continue now.
So the injected mouse survived. Now comes the last part. This time, Griffith injected a mouse with a mixture of both rough strain and heat-killed smooth strain.
شوفوا شو عمل. Last part عمل injection للماوس with both types اللي هو R اللي هو الانفيريولنت avirulent والهيت كيلد استرين، مش الفيرولنت، الهيت كيلد الميتة. He injected the mouse with both. What do you think happened?
Miss,
مع الهيت كيلد. Yes محمد.
Nothing.
He stay alive?
Miss, yes, he's alive.
Okay. مين عنده رأي ثاني? Who has another opinion?
Miss, the mouse dies.
صح، وهذا اللي صار فعلا. هذا اللي صار فعلا. هو استغرب. He was يعني surprised إنه لما كان عندي الR killed didn't kill the mouse. والheat alone didn't kill the mouse. But when he mixed them together, the mouse killed. Okay, what do you think happened? شو رأيكم أنتم؟ Why this happened? في حدا عنده أي opinion؟ يستدير.
Miss,
because it was too strong, like too much for the mouse to handle.
Okay, what else?
Miss, ممكن
والأسرين بتكون violent يعني deadly.
بس ممكن يكون الرف سترين مس ما هو البكتيريا بتقدر تركب أجسام، فممكن تكون دخلت جوا الجسم تبع الكلب.
Let's see what happened. شو كيف فسرها؟ Let's see what.
Strain. That's right. A mixture of the rough strain and the heat-killed smooth strain. Now, what do you think would have happened? Most of us would expect the mouse to survive because both are individually ineffective or avirulent.
يعني هم الاثنين مع كل واحد لحاله ineffective. هذا avirulent وهذا avirulent when you inject them alone. But when he mixed them together and inject them both in the mouse, they become virulent together. How?
However, to his astonishment, the mouse did not survive. How did this happen? And what do you think Griffith concluded? With this, he concluded that there is something from the S strain that was passed on to the R strain, resulting it to turn virulent. Also,
إذا هذا كان الكونكلوجن تبعه.
زي ما أنا حكيت.
Yes. إنه في عندي there is a material من ال S strain اللي هي ال kill strain, okay, was transformed تحولت ال R strain ل virulent. كان avirulent صار virulent. فهو اكتشف transforming material. في عندي material that transform. Okay?
do you know what he recovered from the body of the mouse? He recovered live S strain. Surprising, isn't it?
لما حط أنا عندي الهيت كيلد سترين مع الار سترين هذا الماوس died. We found out إنه صاروا الاس سترين alive. عايشين وشو ميتين هون؟ كيف؟ لأنه في عندي أنا هاي الماتيريال اللي بالاس سترين اللي هي الكلب was moved to the R strain and transformed تحولتها لvirulent. Okay?
This further confirmed that the R strain was transformed into the virulent S strain.
تحولت في material transformed the R2 من avirulent to virulent.
He named the transforming factor as the transforming principle.
وهذا كان اكتشافه. هو اكتشف the transforming principle, which means that there is a material can transform to change one strain from avirulent to virulent. هاي هي the transforming material. إذا شو هو البرنسبل تبعه؟ Transforming principle.
He did not really know what exactly this was and hence named it just the transforming principle.
يعني هو مو عارف، ما عارف شو هذا اللي صار، ايش هو هذا، بس هو اكتشف او الريزلت تبعته انه there is a transforming material. يسمى هذا الاكتشاف transforming principle، ظاهرة الترانسفورميشن او transforming principle، في تحول بصير عندي، في material عم تتحول. اوكي؟
But he predicted that this could be the genetic material.
بس اتوقع he predicted and ممكن تكون the transforming principle هاي هي genetic material. بس شو هي هاي genetic material؟
Was this really the genetic material? Or was it something else? Let's find that out in the next part.
إذا لهون Griff's work stops. Griff's experiment give us a conclusion or give them a conclusion إنه there is a transforming material that transform one strain from avirulent strain to virulent strain or from non-infectious to infectious strain. بس لهون وصل. هو كان يقول he predicted إنه ممكن تكون هاي genetic material but he is not certain. بس يتأكد منه إنه في عندي transforming material أو transforming principle. لهون واضح؟
Miss, can I talk?
Sorry?
Miss,
Yes, talk.
Miss, like it likes the virus when he stands on the cell and puts his genetic material in it.
هذا هنا لا هذا شيء ثاني نعمله بعدين. هلا خلينا نركز احنا بالترانسفورمينج برنسبل. هو بيشبهه بس خلينا هلا نركز بقى هنحكي شيء ثاني عن الفيروس، اوكي؟ إذا هو كان summarize هيك راح I would choose not only one in turn. Who can summarize? What was the first step that Griffith did in his experiment? يلا يا عبد العزيز.
Yes, miss.
عبد العزيز يلا.
First thing, there is a scientist. He want to see that S strain and he want to do an experiment. S strain and R strain. Smooth strain and rough strain.
Bravo.
He make a scientist in a rat.
مين اللي infectious
Yes.
أو harmful؟ الS ولا الR؟
The smooth strain is the harmful.
Bravo. يلا. What is the first step he did?
He bring a mouse and he put the S strain.
Inject. بنقول inject حقنه.
He inject the S strain. And he died. The mouse died.
Thank you. Thank you very much. Murad, continue. Next.
Okay. After he injected the mouse with the S strain and it died, he injected another mouse with the R strain and it lived.
And stay alive, survive. Thank you. Sadil, what next happened? Nujain.
No, miss. So then they.
Yalla Sadil.
Okay, so after they did the SNR, they tried doing. There's one the heat killed the S bacteria, the heat one, which is a virulent, and then there's one where they did the R and S together, and it became deadly because the S.
يا سديد ما تستعجل شوي. خلينا هلا. He used the heat killed strain. What happened after he injected?
It was harmless.
Killed strain. Sorry. What happened to the
harmless.
mouse? Yeah, what happened to the mouse?
It didn't die, it lived.
Bravo, thank you. Jane, next.
The last step was he injected the mouse with the heat-killed S strain and the R strain together,
Bravo.
which caused to the death of the mouse because later on he discovered after the mouse died that the S strain was alive again.
Okay, what did he conclude, Abdul Aziz? You have a question, Abdul Aziz?
Yes, he said that this is a transforming principle.
يعني عندي material, transforming material that حولت transform virulent اللي هو الR ل virulent.
Yes.
But did he know what is this material?
No, he just.
هو بس he found out that there is a transforming material or transforming principle. He predicted انه maybe it's a genetic material, but he was not sure. لسه مش متأكد. هلا who comes after him? Let's see who comes after him and what did they discover.
Imagine we're given a bag full of flowers.
هلا في عنده حدا سؤال على هذا اللي قبل؟ مراد, you have any question? محمد, أي حدا؟
No, miss.
تمام. Let's continue. هلا إذا Griffith he found out that there is a transforming material, but after him comes another group of scientists, and what did they find out? Let's see.
And we are told that there is one flower which has the Midas effect. Anything that the flower touches gets transformed into gold.
يعني عم بعطيكم example. هون عندكم bunch of flowers, and you know that there is one of these flowers that have magic effect. Okay, it can transform things. This is like the transforming material. Okay,
but
how you want to know, and you want to know which flower is the one that has this effect. What you are going to do? Then come bunch of flowers, and you want to know which one of them will make the magic effect. What you are going to do?
Miss, to let them touch things, to turn it to go.
Yes, you will start to touch things. Jane, you have any other idea?
No, miss.
Okay, so you will start to see which one is have the magical effect by touching things. Okay.
Will we find out which flower is the special one? All the flowers look more or less the same. Well, the only thing we can do is make all the flowers touch some object. Isn't that right? So examining every individual flower is the only option we have.
So to know which flower you have to examine. Make examination for this flower to see if it have the magic or not. نفس الشيء. After Griffith came a scientist or a geneticist called Avery. Avery كان له colleague, group of friends. They wanted to examine which material in this cell make this transformation effect. فهمتوا الفكرة؟ فهلا احنا عندنا Griffith he found out انه في transforming material. طب انا جوا السيل عندي كثير materials، عندي carbohydrates وعندي proteins وعندي lipids وعندي nucleic acid، صح؟ They want to know which one has this magic effect, which one has the transforming effect. Okay?
The story was similar in the case of transforming principle. After Griffith concluded his experiment on the bacteria named Streptococcus pneumoniae, the science world was now curious to know more about the transforming principle. Finding the answers was not a piece of cake, though. It was almost 2 decades later that 3 scientists figured out what this transforming principle was.
إذن after Griffith came 3 scientists who بيسموهم Avery and his colleagues. Okay? They found out what is the transforming principle. They want to examine which material that makes this transforming effect.
They tried finding out its chemical nature by testing every single component of the virulent bacterial strain.
أخذوا virulent bacteria. تمام? They want to see, to examine or to test the components. بدهم يشوفوا هاي البكتيريا what are the components that exist inside it or make this bacteria. تمام؟ They want to test each component. Do you understand this point, guys?
Yes.
تمام. All right.
Let's move a step ahead in the timeline and understand the interesting experiment performed by Avery, MacLeod, and McCarty around the year 1944. The three scientists were quite fascinated with Griffith's work. Just like others, even they were curious to know what the transforming principle was. So they thought of continuing the same experiment with better research. Griffith concluded his experiment by inferring that there is some factor that's passed from heat-killed smooth strain to the rough strain, which is avirulent. This factor transforms the avirulent strain to the virulent strain.
هذا كان الكونكلوجن تبع جريفيث انه في عندي material or factor moved from the heat killed to the avirulent R strain and transform it to S strain. هذا كان conclusion لجريفيث.
So the three scientists thought of planning the experiment by targeting the heat-killed smooth strain. Just like a bag filled with flowers, the smooth strain cells had various chemical components in them. And just like how we had to examine the touch of individual flowers, the scientists mixed every single chemical component individually with the rough strain.
نفس الشيء. كيف احنا عملنا هون بالفلاور؟ We take each type and we touch it with the cup to see the effect. نفس الشيء. They take all the components from this strain. أخذوا إيش هي components؟ أخذوا البروتين. They isolated يعني فرقوا الكومبوننتس عن بعض. Okay؟ أخذوا من ال strain البروتين isolated لحال والكربوهيدرات والإنزيمز والDNA. تمام؟ They take all مش إحنا عندي في كل living organism في عندي inside of DNA. أخذوا كل الكومبوننت و they mix each component كل component مع ال R strain عشان يشوفوا which component transform the R strain. You understand what did they do? Guys?
Yes, teacher. I understood.
إذن كمان مرة they isolated the component of the S strain and they mixed each component with the R strain. Tamam.
So let's say this smooth strain cell had these four components A, B, C, and D in it. They mixed the component A with the rough strain and injected it in a live mouse. Similarly, they mixed all the other components individually with the rough strains and injected the mixtures in.
أخذوا كل component لحال من الstrain and mixed it with the R, then inject it to the mouse. What did they find
different
out?
Mice. What they found at the end was only one of those components among all was effective in transforming the rough strain. It was the case in which the mouse died.
شايفين؟ لأنه واحدة من هذول الكومبوننت هي اللي resulting in the infections of the mouse and the mouse died. Okay? Now they want to see what is this component. شو هي هاي طيب الكومبوننت؟ What is it? They make experiment.
The conclusion was that component D transformed the R strain. They isolated this component from the heat-killed strain and tested it further. On chemical analysis, they could figure out that the molecules had a chemical composition similar to that of DNA.
بعد ما عملوا لها examination لقوا إنه هاي الcomponent that caused the mouse to die هي عندها similar composition للDNA. احنا بنعرف إنه الفايف carbon based molecules واحدة منهم الDNA اللي هو nucleic acid. لقوا الكومبوننت تبعها هي زي الكومبوننت تبع الDNA.
Deoxyribonucleic acid, abbreviated as DNA, was known to people by this time, but it was not given much importance. Nobody assumed that it could be such a vital part of the cell. In fact, people thought that proteins made up the cells. And it was also...
They thought before, هي شوفوا كم component فيها. They thought before إنه the proteins هم make up the cell, هم transforming material. Okay? But they concluded إنه the components of the material that transformed the R strain to virulent strain هي DNA after examining its components.
Also believed that proteins make up the genetic material.
هيك كانوا مفكرين. They believe إنه البروتين هو genetic material. بس لا اكتشفوا إنه مش البروتين هو genetic material. DNA هو genetic material. nucleic acid يعني.
This experiment was a huge breakthrough in genetics.
طبعاً.
Many tests were carried out to investigate this further, and the chemical composition of this transforming principle was found. It had components in the ratio that were found in the DNA molecules.
لقوا إنه الcomponents that make this factor هي الcomponents of the DNA itself. فعرفوا إنه ال DNA هي genetic material.
To further understand its nature, this component was tested using chemicals that break down the constituents. For example, certain enzymes like protease degrade protein.
مثلا عندي أنا في ال DNA في عندي إنزيم رح تتعلموا عنه بعدين إنه هذا الإنزيم لو حطيته مع ال DNA يعمل بدنا نحطه على البروتين صار يعمل degradation للبروتين بيكسر البروتين أوكي فحطوا على ال DNA nothing happened حطوا على البروتين لا صار له degradation فهم test they use some chemicals to know the components of DNA اوكي لقوا انه مثلا DNA فيه سلفر البروتين عاده ما فيه سلفر فthese experiments
use of such chemicals help us identify the constituents in a given sample as they will degrade specific molecules only. So in this case, the chemicals used to degrade DNA were used along with others, and it was found that the chemicals successfully
حطوا انه في عندي chemicals that degrade DNA زي enzymes بتكسر ال DNA هو الوحيد اللي كسر ال DNA. chemicals to degrade sugars, chemicals to degrade proteins هذا التفاصيل مش كثير مهمة بس انه بدهم تعرفوا انه استعملوا chemicals to know the components of the factor and they found out it's made of DNA.
Successfully broke down the molecules. It implied that the molecules were that of DNA. This chemical analysis helped in understanding that the transforming principle was nothing else but DNA. And this wasn't easily accepted as a fact.
A few years later, another experiment helped confirm the fact that it's indeed the DNA which is transforming principle. Let's find out more about the experiment.
بس الفيديو راح